So in this question number 1, A, name the structures labeled A to D. So figure 1.1, right, is actually a plan diagram. So when you label the name of A, B, and C, it must be the name of the tissue. Okay, so A is endothelium. So please mark your answer now. A is endothelium. Okay. Do you need to write ciliated? No need to write ciliated. So as long as you've got endothelium, okay, no need ciliated. B is smooth muscle. So as long as you got a muscle, you get the mark. B is smooth muscle. C is cartilage. So you can write C-shaped cartilage, or you can write cartilage also can. Okay. So D is goblet cell or mucus. So look at D here. D is pointing to this cell, which is a goblet cell. It is also pointing into the mucin droplet inside the goblet cell. So I will also accept if you write mucin droplet, because it's pointing to one droplet. Okay, so goblet cell, mucus, or mucin droplet. Okay, part two, you need to show the working. So you're asked to calculate the magnification of figure 1.2 1 mark. Now, because it depends on whether you print it out or you do it on your laptop or you do it on the screen, okay, so your answers could be different. So what I'm looking for is your calculation and conversion of the units must be there. Okay, so therefore magnification M is equals to O over object over A. Okay, O over A. So O is the object. So object here. Now you see there's a straight line being drawn here. So you need to put your ruler over here and measure the length of this line using a ruler, because this measurement given here is the A, the actual height of the cell. So it's 80 micrometer. So therefore you need to use your ruler to measure here. And you need to measure very accurately from this line to this line. So if you look at the mark scheme, okay, mark scheme is M equals to O over A. A is given as 80 micrometer. So your O using your ruler, you can see that the answers that they accept is only a small range. So it's between 2.8 to 2.95. So they accept answers 2.8, 2.85, 2.9, 2.95 only, cm. So you need to write cm here, and you need to write micrometer here, and you need to show the conversion of cm to micrometer. You need to convert cm to micrometer first. So your conversion must be correct. So decicenti, centi is 10 power negative 2. So this one you must memorize, correct? And then micrometer, decicenti milli micro is 10 power negative 6. So when you convert centimeter to micrometer, centimeter is a bigger unit than the micrometer. So when you convert, you take the difference between these two. Okay, so the difference between -6 and -2 is actually 4. So you need to decide whether this is a +4 or -4. So if this unit is bigger than this unit, then it is a +4. So therefore it's 2.8 times 10 power of 4 micrometer. So you need to know how to do conversion, right? So over here, once you've done this question, if you know that you do not know how to do conversion, you must revise and make sure you learn it up, okay? Right, so next question, B. Describe the functions in the trachea of structure A, 3 marks. So structure A is epithelium. So you know that in figure 1.2, in the epithelium, there are 2 types of cells. So you can mention the function of these 2 types of cells. So you can mention about function of ciliated epithelial cell and the function of the goblet cells. So that will give you the 3 marks. So let's look at the mark scheme. Okay, so cilia, so we start with the ciliated epithelial cell function. The cilia is to move, or you can use the word is to sweep. Okay, is to sweep the mucus. So sweep the mucus where? Upwards or towards the pharynx or mouth to be swallowed or sneezed or spat. Okay. So you need to describe fully. So if you say to sweep the mucus, you won't get any mark. It has to be detailed enough. Sweep it to where? Sweep it to the pharynx. Sweep it up towards the pharynx so that you can swallow, sneeze, or spit it out. Okay. And then next we have got goblet cells. Function is to secrete mucus. Okay. Of course, you will not write like mucus is a solution of mucin droplets or glycoproteins. So the function of the mucus is to trap dust or spores or bacteria or any other word with the same effect. So if you say trap air particles, you will not get any mark because you need to elaborate. So what type of air particles that we want the mucus to trap? So you need to say dust or spores, bacteria, viruses, fungi, and so on. Pollen grains. Okay. So prevent the air particles, in order to prevent air particles from reaching the alveoli or bronchi or bronchiole. So that's why we want to trap it. We don't want it to reach to alveoli, bronchi, bronchiole, so as to reduce the likelihood of infection. Because when the bacteria and viruses are able to enter alveoli, bronchi, bronchiole, they can infect the cells of these airways and then cause diseases. Okay. So this is the detail that when you study biology, you need to know. So I highly encourage you to do the quiz to test yourself whether you have mastered every chapter. All right. So part 2, describe the function of structure C. So C is the C-shaped cartilage. So you need to say that this is to ensure that it will keep the trachea open, or it stops the trachea from collapsing. It stops the trachea from being compressed. So it will stop the trachea from, it reduces the resistance to airflow by maintaining the lumen to always be open in the trachea. So that means you need to talk about trachea remains open. Okay. Okay, next question. So in this question, someone actually asked me, is this already not in the syllabus? Even though this disease is not in the syllabus, but the nature of the question can be asked in exam because it's very straightforward and it covers the topic that you have learned. So whatever question I've given here, I've already modified from original question so that you only ask what you need to know rather than what you have not learned about the disease. Okay, so figure 2.1a and b shows diagrams drawn from section X of lung tissue. So it's a section from the lung. A is from a normal person and B is from a person with a lung with a person who suffers from emphysema. So whatever is the disease, so the structure of the lung will look like in figure B. So A state two differences visible, means you can see, okay, in the diagrams between the normal lung and the lung of the emphysema sufferer. So you must base on the diagram to describe. So that is straightforward. So first you can see that in B there are less number of alveoli, correct? And then there are more air spaces. So these are the two answers. First one is there are less alveoli, or you can say that less alveolar wall. And the second one is there are more air spaces. And of course, when you got less alveolar wall, there will be less surface area for gaseous exchange. So that is a possible third answer. Less surface area for gaseous exchange. Okay. So B, with reference to figure 2.1, explain why people who have smoked heavily for a long time often have difficulty in breathing and obtaining sufficient oxygen. Breathing and obtaining sufficient oxygen based on the question, I will say that this one is a suggest question because you have not learned about the disease. So based on the diagram, so you can see that part A, the answer in part A will lead you to answer the question in part B. Because in part A, we already say that there are less number of alveoli, or there are less surface area for gaseous exchange, right? So therefore, this answer will link to part B. So why is it this person cannot get sufficient oxygen? It's because there are less, so your answer will be either you say less surface area for gaseous exchange, or you can say that there are less number of alveoli for gaseous exchange. You can also say there are less number of capillary for gaseous exchange. It's the same. Because when the wall is missing, you already know that the wall also contains capillary. So when you don't have the wall, you will not have the capillary as well. So less number of alveoli, less number of capillary, less surface area for gaseous exchange. Therefore, this person cannot get enough oxygen. Okay? So C, some alveolar cells produce a surfactant. Now, even though C is talking about the alveoli, you can see that the nature of this question is asking you about cell signaling. So as you read the passage, you need to visualize or draw out what the passage is trying to tell you. Then only you can answer the question here. Okay. So some alveolar cells produce a surfactant that helps to prevent the collapse of alveoli on exhalation. Too much surfactant will decrease the efficiency of gas exchange in the alveoli. So glycoprotein known as GM-CSF is released by some cells of the immune system. So cell of the immune system can secrete GM-CSF when there's too much surfactant in the alveoli. So over here, you know that when you learn about cell signaling, in the first thing is you got the stimulus. It's actually based on stimulus response model. Receptor, right? Transmission. So this is a diagram that you must always remember. Then effector, and then response. So this is a stimulus response model that you have learned in your secondary school. So whenever in a cell signaling, it must always start with a stimulus first. So stimulus is a change from the norm, changes from the norm. So for example, temperature changes, blood glucose concentration changes, pressure in the surrounding that changes, partial pressure of oxygen in the surrounding that changes, pH that changes. So change from the norm. So over here, what is a change from the norm is actually too much surfactant being secreted in the alveoli. So the too much surfactant is not the norm. Norm means normal, not the normal level. So it acts as a stimulus. Who will it stimulate? So usually receptor belongs to a cell. So it stimulates the cell, which is actually the cell of the immune system. So what does a cell do? So you see that after transmission, I think, sorry, this is not transmission, it's in bracket. So transmission will go to a coordinator, okay, or the control center. So transmission, so after the cell of the immune system, which act as a receptor, which can detect this change, it will then send a message. So whenever you say transmission, it is trying to send a message. So this message you learned already is either chemical, this message is transmitted by the nervous system or the endocrine system. Okay, so this is a revision, okay, because I have already taught you this one before. So this is a revision. Okay, so either nervous system or endocrine system. So transmission of the message is by nervous system or endocrine system. So nervous system is to transmit using the nerve cell by electrical impulse. So in the cell signaling, you learn that chemicals are being released, which are called the hormones, right? Hormones. And hormones can act as the chemical messenger. Chemical messenger. So it is a molecule that carries a message. So here, the messenger molecule is the GM-CSF. It is secreted by the cells of immune system. Okay. So after that, it will be transmitted. So this transmission, and the effector is which cell? So receptors, so that the effector cell, so the cell that is target, the target cell of the message, the chemical messenger. So chemical messenger will bind to the target cell. So target cell must contain receptor for CMGFF. It must contain receptor for the, GM-CSF. So these receptors are found on the cell surface membrane of alveolar macrophage. So that means the target cell is alveolar macrophage. So this is the target cell of the messenger, target cell. Because target cell has got receptor for this messenger. So this is what the passage is talking about. So you have to relate the passage to the basic information you have learned. So the question asks the student to explain how the correct quantity of surfactant in alveoli is the result of a cell signaling mechanism. And this is 4 marks. So you already learned from cell signaling mechanism, you need to first state what is a cell that will secrete the signal, what is a signal molecule, and what happened to the signal molecule and then after that, in the target cell, what happened to the target cell? So 4 marks. So let's look at the mark scheme. Here, okay. So altogether got 7 correct points. Accept the glycoprotein for GM-CSF throughout. That means to say that if you don't say GM-CSF in your answers, if you write glycoprotein, the examiner said they will accept because in the passage, it actually talks about glycoprotein, right? A glycoprotein known as GM-CSF. So therefore, it's acceptable. So first, there must be a stimulus, right? So the stimulus is excess surfactant that stimulates the immune system cells. So after that, the cell will release or secrete GM-CSF, ignore produce, because the cell must produce GM-CSF, and after that, it must be secreted out of the cell. So a better word is secretion. Then the GM-CSF act as a signal molecule. So act as a signal or a signaling molecule or a messenger. So on the right side, it says messaging molecule or messenger molecule. Okay. So pay attention to the word that they use to describe. Number 4, so this signal molecule binds to the receptor on the cell surface membrane of the alveolar macrophage. So keyword is receptor. And then number 5, refer to specificity. So by now you already know specificity has to do with the shape. So it's talking about GM-CSF can bind to receptor by complementary shape. Okay. So by now you need to be very careful because you also learn about complementary base pairing. So complementary base pairing only applies in DNA as well as RNA. So during DNA replication and transcription. So this is complementary base pairing, and also during translation. So during DNA replication, transcription, translation, complementary base pairing is needed. But other than that, this is about binding of a messenger molecule to the receptor of the cell. Is not complementary base pairing because there's no bases there. So there is only complementary shape. So this is something that you have to be very careful when you write your answer. Okay. Number 6, so it leads to, so when the messenger molecule binds to receptor, you know that it will trigger a response in the cell. So what kind of response? So the response I've mentioned just now, stimulus response model. The response is to remove the surfactant because this is a macrophage, right? A macrophage. So it stimulates the macrophage in the alveolus to remove the excess surfactant. Okay, so that is what the response that we are looking for. So therefore, you have to think about stimulus response, stimulus receptor transmission. Over here, there's no coordinator. We got the effector. So the effector is a cell or an organ that will carry out the response. So in this case here, it is a cell, which is the alveolar macrophage, which will carry out the response. So always remember the stimulus response model. That will help you to answer your question more systematically, more crystal clearly. Okay, so one more mark, which is the final mark, is an AVP. So the detail of what happens inside the cell. So you know that when the primary messenger binds to the receptor, you have got a secondary messenger that will be produced inside the cell. Because the primary messenger cannot enter the cell, so it can only bind to receptor, activating secondary messenger molecule to be produced in the cell, and that may activate a series of enzyme cascading reaction. So you know that enzyme cascading reaction function is to amplify the signal. So this is a revision. Amplifies the signal. That means from one signal molecule that arrive, you can, after the enzyme cascading reaction, it can lead to high number of molecules or enzymes being synthesized. Okay, so the response will be high number of whatever product that you want the cell to produce. So signal transduction occurs inside the cell, means transduction is conversion. So conversion of the signal. So the signal is actually this molecule here. Convert to what? So it convert to a series of enzymes that will be produced. And in one of the steps, it involves phosphorylation of the enzyme. So this is actually detailed. Okay, if you not learn, it's okay because you learn this again in your A2. So phosphorylation is detail that you learn in A2. Okay, so a series of phosphorylation will occur, a lot of enzymes will be produced. So this is called signal transduction, conversion of the signal, and it also leads to amplification of the signal in this process here. Okay, so revision of cell signaling for this question. Next, so three is you need to fill in the blank. So even though it talk about lungs, it is actually asking you about cancer, right? Cancer is in the mitotic cell cycle chapter 5. So you need to fill in the blanks, right? I have students who did not do this question. I have students who did not even answer this question. It's blank, blank, blank. Especially the long questions are blank, blank, blank. So next time, please try to answer the question because there's no reason why you tell me you cannot answer. Fill in the blank. If you say this is very long, it's a 4 marks, at least you fill in to get at least 2 marks. No reason is totally empty. Okay, so I checked your submitted answers. Okay, I will check your submitted answers so I know that how you have performed. Right, so make sure you do your part. Okay, don't just say, oh teacher, I don't know how to answer. Then you try. You can refer to your notes because every question that you do is a revision. If you do not know how to answer, that means you do not know those parts. You have to refer to your notes, isn't it? And then try to answer the question, isn't it? That is what you need to do. Not say, teacher, I don't know how to answer. So you don't know how to answer, so what? Then you have to learn, isn't it? Refer to the notes and learn from there because the question will show you whether you know or not that part. So it's an indication for you where you need to revise. So next time, don't submit questions that are not answered. Okay. So let's look at the answer. So lung cancer is the uncontrolled mitosis, or you can say uncontrolled cell division of the cells in one or both lungs. So this is actually defining what is cancer, uncontrolled cell division or uncontrolled mitosis. So these cells do not carry out the functions of normal cells and do not develop into healthy lung tissue. They grow. As they grow, the abnormal cells can form tumors and impact the function of the lung, which is to provide oxygen to the body via the blood. That means to say that when the tumor cell starts to grow in the lung, because it continues to grow bigger and bigger, it may suppress and damage the cells in the surrounding. So that causes the lungs to not to be able to function properly because there could be a less number of alveolar cell. There could be bronchiole and bronchus that are being blocked by the tumor. Okay, and it might even damage the blood vessels, damage the blood vessel wall. Okay, so a cancer begins with an error. So what is the error? It is a mutation in a cell's DNA. Okay, there are over 4,000 chemicals in the cigarette smoke, including 40 known carcinogens. So carcinogens are agents. You see, it is given here. You need to give the name to this definition. A substance capable of causing cancer is carcinogen. So for instance, oncogene is a genetic material that carries the ability to induce cancer, right? So you already learned the only term that you learn for a genetic material that can cause cancer is called oncogene. So it's testing your understanding of the term used to define something. Okay, so induced cancer can be developed due to cigarette smoke. It is a sequence of DNA that has been altered from its original form. So the original form of oncogene is called the proto-oncogene. Okay. So next, beside tumor suppressor genes might be switched off or inactivated. So you know that only the gene can be switched off. So this part is talking about the gene. So here also continue to talk about the gene. So you need to name the gene. Might be switched off. So whenever this gene is switched off, it will lead to cancer. So what gene is this? So second gene that you learn is tumor suppressor gene. So gene that suppress tumor formation might be switched off, leading to cancer. So the function of the gene is to slow down cell division, because when you switch off the tumor, that means you switch off cell division. So you prevent tumor formation. Okay, so it may slow down cell division. It may also repair the DNA so that tumor cannot be formed. So there are many ways to prevent tumor from forming. One is to slow down cell division. Secondly is you go right to the root cause. Whenever there's a mutation, then we try to repair the DNA to convert the mutation back to the normal base sequence. Usually, repair of DNA happens immediately after DNA replication. So in the stages of DNA replication, the very last stage that you did not learn is there will be an enzyme that will move along the DNA to check whether there's any mistake. It means any wrong base that is being added in, and the wrong base will be removed and replaced by the correct base. So this is the last step in DNA replication. So this is called repairing the DNA. So if it's not repaired, that means the mechanism of repairing DNA is impaired. It's not functioning well in the cell, okay? Or it leads to apoptosis, so it's already placed in bracket, programmed cell death. So what is a biological term for programmed cell death? Is called apoptosis. So, which means to say that if you look at the past year questions, sometimes this nature of question will come up. That means there's a passage with empty spaces. You need to fill in the blank. So when you need to fill in the blank, which means to say that if you choose not to learn the word, for example, apoptosis, you will not be able to answer this question. If you choose not to learn the word oncogenes, proto-oncogenes, tumor suppressor gene, carcinogen, then you may not be able to answer this question to get the full marks. So therefore, it is very important that you have sufficient time to learn up everything that you have learned in your syllabus. Everything that you have learned in the chapter, you need to give yourself ample of time to learn them up. Okay? Right, next question. Question 4. So question 4 is a very good question because it is a graph. So you need to be able to describe a graph is one of the skill that you need to be able to do. And I guarantee 100% that this kind of question will come out in exam paper. 100%. You open any past year paper 2, surely there is one or else 2 questions that ask you to describe the graph. So you need to learn how to describe the graph to score full marks. Okay, so you need to practice. So therefore, when you are preparing question for revision in the subsequent days to come, if you come across a question with a graph like this, it is actually good for you to choose that question. So at least you can practice describing and explaining the graph, especially when it comes to enzyme chapter. Enzyme chapter got a lot of graphs, okay? Infectious disease chapter also got a lot of graphs, okay? So let's do the question together. So figure 3.1 shows the mean yield of tar and nicotine and carbon monoxide for British cigarettes from 1935 to 1982. Detailed annual figures are available from 1969. So that means from 1969 onwards, there are a lot of detailed figures. That's why there are more points on the graph versus the timing before that, you've got less data available. And also take note that in this particular question, we have got 2 y-axis. the one on the left and the one on the right. So therefore, when you are reading for tar and carbon monoxide, you have to read this y-axis. But when you're reading nicotine yield, you need to read from the y-axis on the right side. So that is the catch that you need to be careful of. Okay, so let's look at the question. Compare changes in the tar and nicotine yields of British cigarettes between 1950 and 1980. So first, you need to know exactly where you need to describe. It's 1950 to 1980 only. So don't describe any other parts of the graph. And only describe tar and nicotine yields. So therefore, we go back to the graph to look for tar and nicotine yields. That means we only describe tar, which we look at this side, and nicotine yield, which we will look at this y-axis here. Okay, so every time when you want to describe a graph, first step is you give the general pattern. I already mentioned general pattern for both of them. Then you can talk about the gradient, talk about the interception if there's any, okay, and you need to quote figures, a pair of figures. A pair of figures means you need to quote from X to Y axis. It changes to this X value and Y value. So this is called a pair of figures. Okay, so this is X1. This is Y1. It changes to X2, Y2. And remember to indicate the unit. Okay, so general pattern you can see for tar is going down. Tar is going down. Okay, and nicotine is also overall, it is also going down overall. Of course, let me see. Yes, it actually goes down because we need to start from 1950. So starting from 1950, overall going down. But you look at the tar, it actually goes up, down, and then this is slightly down. So generally is going down. Okay, generally for both of them, it is going down. So this is the first statement. So for both tar and nicotine, the mean yield decreases from 1950 to 1980. So the first statement already mentioned. Completed. Now, secondly is the detail. So before we move on to detail, we need to check how many marks is given for this question. What is happening? Okay, 2 marks only. So I already obtained 1 mark for general statement. So for the second mark, it's very easy. I have to quote figures. Because every time when asked to describe a graph, there will always be mark given to quote the figures. So since I already obtained the first mark, so now I describe the second mark. So second mark is very easy. Second mark, you need to indicate a pair of figures for tar and nicotine, both of them, from 1950. So you need to read accurately from here. What is this value? The first one is wrong. Huh? This one must be totally vertical. Totally vertical. Am I totally vertical? Why 1950 is not 1951? Yes, this is totally vertical. Correct. Okay. So nicotine, you must read from here. Then you read on 1980, which is the last value, right? Because they ask you 1980, so you don't read the last point. You read at 1980. What is the value here? So make sure that this horizontal line is parallel to the x-axis, and tar is this value here. So you read, okay? So you have to read very accurately. So you said that for tar, from 1950 to 1980. So I already given the value, which is actually from x1 to x2. Correct? So this is from x1 to x2. So now I need to put the y1 and y2. So for ta yield, okay? This is mean ta yield, which is the y-axis, decreases. Then you have to give me value from what value to what value. Okay, so therefore, Amar, can you give me the value for ta? Decreases from what value to what value? So 1.6. And then unit, please. Unit, please.
mg nicotine, mg per nicotine.
mg per cigarette. Okay, cigarette. So whenever you label y-axis and x-axis, this is called label. Nicotine U is the label, stroke. After the stroke is the unit. So the unit is milligram per cigarette. Same here, this is label. Tar and carbon monoxide mean U is the label. After the stroke is the unit. So this is how you need to label the y-axis and x-axis in exam is also this way. When you're asked to draw a graph, label it, put the stroke, behind the stroke, put the unit. So usually you copy this whole thing from the table of the data. You copy this whole thing from the heading of the table of the data. So meaning to say that you got a table, right? Usually from the table, you draw the graph. So in the table, first column is usually the independent variable. So you copy this heading to your x-axis. Together with the unit, you copy this heading to your y-axis because this is dependent variable. This is independent variable. So your format of graph is also x-axis is independent variable, y-axis is dependent variable. It must always be like this or else you lose 1 mark in exam for the wrong orientation of the axis. Okay. All right, so this is how you describe the graph. So let me recap. Number 1, you have to give a general, state the general pattern. Number 2, you need to, in this case here, quote a pair of figures. So you have learned to code the figures correctly together with the units from the correct y-axis. Okay. So that's all for this question here.